Hello dear students, how are you? In the last class we have discussed the second lesson, excretion, and in excretion what we have discussed, the classification of animals on the basis of excretory products that are ammonotelic, ureotelic and ureotelic. So the ammonotelic animals, they excrete ammonia, and uricotelic animals excrete uric acid, and ureotelic they excrete urea. We have already discussed this one, and we will move on to the next topic, that is the structure and function of nephron. So what is a nephron? Nephron is the functional unit of the kidney. Each kidney consists approximately 1.2 million nephrons. So the nephron is the structural and functional unit of a kidney, which is responsible for the perfect filtration of the blood and formation of urine. That means the elimination of excretory waste, poisonous substance from the body is eliminated through this functional unit nephron only. So this nephron, how many nephrons are present in each kidney? More than 1.2 million nephrons are present in each kidney. So, are there any functional differences, any types in the nephrons? Yes, there are two types. According to their relative position in the cortex, do you know what is cortex? Cortex and medulla, these are the two parts of the kidneys. So these nephrons are present in the cortex, and in this cortex the relative position, the nephrons are of two types. One is cortical nephrons or superficial nephrons. Superficial nephron is made up of superficial glomeruli which occupy nearly 2/3, that is 85% of the total number of nephrons are made up of this type of cortical nephrons. And the second one is juxtamedullary nephrons. These juxtamedullary nephrons, they are 1/3 of the cortex, and they constitute about 15% of the total number. And here you can see some of the differences also. They are almost similar in a small in the first one, cortical nephrons are small in size, whereas juxtamedullary nephrons are large in size, and Henle's loops are very short in cortical, whereas in juxtamedullary Henle's loop are very long and extending. So these are the primary differences between these two nephrons. When we see the structure of a nephron, each nephron consists of 6 successive parts. What are they? The first one is renal or Malpighian corpuscle. Second one is proximal convoluted tubule. Then third is thin segment loop of Henle, that is Henle's loop. Fourth one is proximal straight tubule. Fifth one distal straight tubule, and sixth one distal convoluted tubule. See, here you can see PCT is proximal convoluted tubule and DCT is distal convoluted tubule. Let us see. See, here is the difference, size difference. Here is the size difference. This one is the juxtamedullary nephron, and this one is the cortical nephron. This is the size difference among these two things. And when we move to the structure, renal corpuscle and renal tubules, these are the main two parts. Again, renal corpuscle is divided into Bowman's capsule and glomerulus, and renal tubule is divided into proximal convoluted tubule or PCT, loop of Henle or Henle's loop, then DCT or distal convoluted tubule. So when we see the detailed study of the structure of nephron, the first foremost, the renal capsule is very important, and this is the head of the nephron. This also known as renal or Malpighian corpuscle. Malpighian corpuscle or renal corpuscle measures in diameter about 200 microns, and it is divided into two parts, that is glomerulus and Bowman's capsule. So when you see here, this is the structure of the renal corpuscle. You can see here a glomerulus and a Bowman's capsule here, and the glomerulus is surrounded by, see here, the glomerulus is the capillary tuft, that is this glomerulus is formed by the afferent arteriole and efferent arteriole. These two arterioles are the blood vessels, and they form a capillary tuft like structure which is known as Bowman's capsule, and the afferent arteriole breaks up into 50 capillary loops and forms the glomerular tuft which lies within the double-walled sac-like Bowman's capsule. At the beginning of each nephron, a tuft of 6 to 8 renal blood capillaries invaginated into the end of the tubule. This structure is known as glomerulus. Glomerulus is nothing but blood capillary tuft. There are about 20 to 50 capillary loops present in the glomerulus, which make up freely branching anastomotic network. Just before entering the glomerulus, the media of the afferent arteriole is found to contain a thick cuff of large modified muscles called as juxtaglomerular cells. So this is glomerulus, nothing but a blood capillary tuft made from afferent and efferent arterioles. And here in this glomerulus, from the blood, the extra water and other salts and waste, nitrogenous waste are extracted from the blood. So that is glomerulus. And what is Bowman's capsule? This is the second part present in the renal or Malpighian corpuscle. So Bowman's capsule, it is a cup-shaped structure with outer parietal layer. So what is this? It is a cup-shaped structure with outer parietal layer and inner visceral layer. So these are two layers. Outer one is known as parietal layer and inner one is known as visceral layer. The parietal layer is made up of typical squamous epithelium of flat polygonal cells, and visceral layer is made up of large cells known as podocyte cells. So here you can see the inner structure, podocyte lining. So these are the podocytes, the endothelial cells. Here, what is the function of this Bowman's capsule? As we have already discussed, it is a double-walled cup-shaped structure. It is a sac that encloses the glomerulus, and it transfers the filtrates from the glomerulus to the proximal convoluted tube. That is the first part of the renal tubules. So let us move on to the renal tubules. Yes. This Bowman's capsule, what happens? It is ended into a PCT or proximal convoluted tubule or pars convolute. This is also known as pars convolute. This PCT is lined by a single layer of cubical cells, the free borders of which are striated or brush bordered. So here you can see. So here in this picture you can clearly observe the Bowman's capsule's transverse section, and in this you can see the tuft of glomerulus, the blood vessels are ending, and here you can see a parietal layer and a visceral layer, and between these two layers urinary space is present. This is also known as Bowman's space. So this Bowman's capsule, now it leads into PCT. Here you can see this is denoted in green color. It lies in the renal cortex, and PCT is lined by cuboidal epithelial having brush borders with long microvilli for increasing absorptive area, and this results on a basement membrane that reabsorbs most of the useful substances of the filtrates like sodium and water, then bicarbonate, some chlorides and glucose. So this is the PCT, that is proximal convoluted tubule. So in this picture you can see this is the PCT here, proximal convoluted tube. It is present in green color here. So the proximal convoluted tubule, the cells of these tubule are very active metabolically and contains a large number of enzymes also. These cells absorb about 2/3 of the water of the glomerular filtrate, and all the glucose, amino acids and sodium, chlorides and phosphate are to be reabsorbed. This PCT, it will end into a loop of Henle or pars nectar. This is also known as pars nectar. It leads into a Henle's loop. Yes, the second one is loop of Henle or pars nectar. It is a U-shaped loop present in the medulla of the kidney. The loop of Henle can be divided into three parts, that is descending limb of Henle's loop. Here you can see that is the descending limb of Henle's loop, then thin walled ascending limb of Henle's loop, then thick walled ascending limb of Henle's loop. First one is descending, then the other two are ascending. First one is thin walled and the second one is thick walled. So it begins in the cortex, receiving filtrate from the PCT, proximal convoluted tube, and extends into medulla and then returns to the cortex to empty into the DCT or distal convoluted tube. So this is Henle's loop, and so the Henle's loop is also having three parts. Descending limb of the loop of Henle, this is a part of the counter current multiplier, and the second one the thin walled ascending limb. And the first, when we see the descending limb, what are its features, main features? It is a part of counter current multiplier and fully permeable to water and completely impermeable to solutes. So it receives filtrate from the PCT and allows water to be absorbed and passes salty filtrate to the next segment. Then the next part is ascending limb. This ascending limb has two parts, thin walled and thick walled parts. And what are the main functions of this ascending loop? This is the impermeable part to water and actively transports or reabsorbs salt into the interstitial fluid of the pyramids in the medulla. It saves salts and passes the water. So this is the descending limb, and descending limb we have already discussed that it has two parts, a thick walled part and a thin walled part. Thick segment is constituting about 4 or 5th of the descending limb, and it lies both inside cortex and medulla. The cell lining it are cuboidal, and in thin segment it is narrow part of the descending limb, and it lies in the medulla and is lined by flat epithelial cells. Here you can see the descending thick limb and thin part of the Henle's loop. Here you can see this is the glomerulus Bowman's capsule, and this one is the proximal convoluted PCT part, and this is a Henle's loop, and the first one is thick descending limb and this is a thin descending limb. So here you can see the first one is thick part, which is here you can see the thick part, and here you can see the thin part. So These are the thick and thin limbs of Henle's loop, and this Henle's loop, what happens, it again opens to where DCT or distal convoluted tubule. This is DCT and this is PCT. PCT is the first one, that is proximal convoluted tubule. The next part is Henle, and Henle's loop again it opens into distal convoluted tubule. So these are the things you have to remember. And then what happens, this distal convoluted tubule, it opens itself into the renal tubules. So these are the things. Then what we have to discuss, the next last part is distal convoluted tubule or DCT. Distal convoluted tubule or DCT, it is highly coiled part of nephron, and it lies close to the Malpighian body, and it is variably active portion of the nephron, and it is lined by cubical epithelium which is nodded but without any true brush borders. And this DCT what it does, it actually comes in contact with the juxtaglomerular cells, and this connects with the next part of the collection tubule, and it empties all the waste material into the collecting tubule. This is the structure of the nephron. This is the basic structure of a nephron, and here you can see the collecting tubule which is collecting the nitrogenous waste material or urine from the nephron. So what is collecting or straight tubule? Collecting tubule is lined by pale cuboidal cells, and which is about 20 millimeters long. Several collecting tubules from different nephrons. So from one nephron, one collecting tubule arises, and different collecting tubules from different nephrons are combined to form the duct of Bellini, which opens at the apex of renal pyramid. So these collecting tubules are collectively formed as duct of Bellini, and it is opened into a renal pyramid. I think you remember renal pyramid in your lower classes. Renal pyramid you might have studied. See here in this picture you can see the path of moving of the nitrogenous waste or urine. Here you can see the urine is moving from the PCT proximal tubule, and then it is moving into the Henle's loop, and again it is moving to the distal tubule, and again lastly it is collected by the collecting duct, and it is opened into duct of Bellini, and again it is forming renal pyramid. So these are the functional performance, the functioning of one nephron. And see, these are nephrons which are collecting the internal, and nephrons, what they are doing mainly, the nephron remove urea and uric acid from the blood, and the nephron regulates blood pressure also. The nephron also removes pigments, drugs, poisons, excess vitamins from the blood, and it regulates the amount of mineral salts in the body, and also regulates the blood pH. That's why these nephrons are very, very important, and you have to learn it well. And it is also an important question for your final exams. Let us recap the main functions of the kidney. What are the functions of kidney? It excretes waste products, especially the nitrogenous and sulfur products containing end product of protein metabolism, and it helps to maintain the normal hydrogen ion concentration, that is pH, of the body fluids and electrolytes. It helps to maintain water balance of the body. Please make a note, take a book, a pen. Please listen carefully and write down. It helps to maintain water balance of the body and thereby the plasma volume. It helps to maintain the optimum concentration of certain constituents of blood, that is selective reabsorption is going on here. That's why the blood pH and the blood thickness is maintained. It eliminates drugs and various toxic substances, poisons from the body, and it manufactures certain new substances, ammonia, hippuric acid and inorganic phosphates. Ammonia helps in preserving acid-base equilibrium. It helps in maintaining the osmotic pressure in blood and tissues. It helps in the regulation of blood pressures during hypoxia, in condition of emergency through the liberation of renin from the juxtaglomerular apparatus, and it helps in the regulation of erythropoiesis through the formation of erythropoietin, and it also plays an important role in vitamin D metabolism. So kidneys are very important, and in each kidney the nephron is the functional and structural unit of kidney, which is performing the main basic function of filtration in our body. And this is an important essay question, or it may come for a short answer question asking the functions of, they may ask like this, they just only ask about the functions of the nephron or kidney, or they may ask the structural elaborative structure of the nephron. So please listen this carefully, make a note, take your book, please buy the books. Then you please go on reading what is discussed in the today's class, and then if you have any doubts, please ask me through a message or WhatsApp. Thank you, students. Thank you and bye. Tomorrow we will meet in the next class and we will discuss the next topic, urine formation. Thank you. Bye.